With the global population aging rapidly, the number of ALS cases worldwide could increase from 222,000 in 2015 to more than 376,000 in 2040, representing an increase of 69%. With no cure and no effective treatment to halt or reverse the progression of this disease, the world is working together to fight against ALS, bringing a wealth of new scientific understanding about the physiology of ALS. What are the innovative targets and pathways that researchers are going after? Why it is important to seek a multidisciplinary approach for ALS treatment? What will the landscape of ALS research and care look like in the near future? In this session, our guest speakers will share their insights. Please welcome Jeffrey Rothstein, Director of Brain Science Institute, Director, Robert Packard Center for ALS Research, Johns Hopkins University; Sabrina Paganoni, Co-Director, Neurological Research Institute at Massachusetts General Hospital, Physician Scientist, Healy and AMG Center for ALS; and Kuldip Dave, SVP of Research, the ALS Association.
Well, thank you for the Wuxi AppTech team for inviting me and Jeff and Sabrina to be on this panel. Maybe let's start with better understanding of the biology. Jeff, Sabrina, this is what we like to call target ID and target validation. And in the past, target ID, target validation was, let's look at cells, let's look at animal models. But now we have tools that are much more human relevant. And so, Jeff, would love to hear your thoughts about, the use of some of these human models to optimize, to make target ID and target validation better.
Yeah, so I agree with you. I think there are newer models. I mean, we spent 2 decades using rodent and fish, and to be clear, these are they have been useful tools, but they're not human. And I think our patients certainly all sort of point out that we haven't as many drugs as we'd like, and why are we using mice? And fortunately, the technology has increased in the last decade. Now we actually do have human models. We have what cancers had for a long time. We have the way of essentially biopsying the nervous system by using blood cells from patients and creating IPS cells, induced pluripotent cells. And these are then turned into our patients' cortical neurons or spinal neurons. And those, some, not all, but many of us believe is sort of a future platform. When you think 10 years in advance, is this the platform that'll teach us more about what's really happening in patients? Sure, it's not a connected brain, but on the other hand, most of our drugs aren't about connected brains, like a disease like epilepsy. They're about what's wrong in a given cell and the cascade of events that goes wrong in a cell. And so these cell paradigms, these new cell paradigms are really being used, or beginning to be used worldwide. The other really important thing about this technology that trumps, in my mind, really many other approaches is that many of the models we've used for the past 25 years have relied on the mutations of ALS, which we know are really important to teach us how the disease begins in that subset of patients. But they don't necessarily teach us what's going on in the vast majority of our patients, sporadic ALS. And since these cells can be built from all of our patients, in fact, they have, one of the programs I'm involved in, Answer ALS, but there are others around the world where we derive these, I tend to call them biopsies, from our sporadic patients. They may give, and I want to underscore may, because I don't know for a fact that they'll be useful, but what I do know is that the mouse has not been as useful as we've hoped. And these will give us a platform that's human-based, that's really based on sporadic ALS. And when I think of the next 10 years, I really think that's what we have to focus on as much as we can, the real population of ALS patients, sporadic patients. And to be clear, it's not the only model system. People are actually taking the cells and sort of rebuilding forms of the brain called organoids, another way of looking at cell-cell interactions, because we know in ALS that's not just the neuron that dies, that there's inflammatory cells called microglia. There's other support cells, glial cells, oligodendroglia and astroglia. And all of these human platforms may give us that window into 2 new ways. I don't want to lecture, so I could stop, but I think that's important.
Yeah, and Jeff, what I'm thinking about is target ID and target validation were really slow. And what I'm hoping that in 10 years is we are quickly able to nominate the targets, validate them or invalidate them and quickly close the door on them. Because, the thing I see in the field, and Sabrina, I'll come to you in a minute here, but, is that it's taking time. And so 10 years from now, if we can use these human systems that you just mentioned, Jeff, to, quickly say, is this target druggable? Is this something we can take into the therapeutic pipeline? And answer that question quickly. I think speed matters. We got to work on the ALS clock, and so that is my hope for the future. Sabrina, coming to you, there's this whole idea about even using patients' own cells as a personalized medicine approach, and would love to hear your thoughts on, as a clinician, how do you see the promise of that 10 years from now?
Yeah, I mean, that would be fantastic. I mean, that follows what Jeff was describing in terms of target validation using cells or material that comes from actual patients, not just relying on specific mutations that can be tested in the mouse model, for example, but really giving us the opportunity to test drugs that potentially can be personalized. I do want to mention that what I think is going to be really important in the future, again, thinking about what comes next, is that as we discover, hopefully, at least a few drugs that have a positive impact, it will be very important to go back to the models and then actually test them in the models and then validate the predictive value of the models, because the truth is that we still don't know which model and obviously the devil is in the details, even when it comes to cells, personalized cells from patients. How do you actually, what's the best way to grow them? What's the best phenotype that you're looking for? So as we develop new drugs, and quite frankly, even with the ones that we have, it would be great to then create almost a battery of tests, we validate the ones that are most predictive, and that could be more than one actually, and that will actually speed up the drug development process.
That's a really good point, Sabrina. And the other thing I think about here is, at ALS Association, I get asked all the time, how many models should we test our drug in? And I always say, I rather you test it in one model that makes the most sense mechanistically as to how your drug works. So if your drug is reducing aggregation, then use a TDP-43 model instead of a SOD1 model, right? Or if your mechanism is targeting mitochondria or oxidative stress, then use the SOD1 model. But I'd rather you use one model and do a much more deeper validation in that one model. Well, do a full dose response. If it's an agonist, antagonist, do a pharmacology study, do PK, instead of trying to do efficacy in 2 or 3 different models. And so I'm so glad you talked about that. Jeff, what's, one of the things that you brought up was inflammation. And, the one thing that you and I have talked about this in the past is, right now, if you look at our ecosystem, 40-50% of drugs that are in the pipeline right now in phase 1, 2, and 3 are neuroinflammation focused. And, do you worry, like I worry, that, we may have all of our eggs in one basket? And do we need to expand more? And does our understanding from omics, from initiatives like Answer ALS, help us expand and diversify the pathways we're looking at?
So there's a lot to that, and I'm going to go through a couple of things quickly. One, so I'm the most gray-haired person here. Trials in the 90s were really when there was a growth of ALS trials, began with some of the growth factors. A huge number of trials in the 1990s were what we as neurologists, especially neuromuscular specialists, ALS is sort of only one of many of our neuromuscular diseases. Many of them were actually autoimmune diseases. And so we were pretty familiar with using anti-inflammatory related pathways. Mm-hmm. And so the 1990s was littered with trials around anti-inflammatory drugs. Obviously, they didn't work because we'd be using them. But to be really careful about that statement, the trials in the 1990s are not as sophisticated as they were today. So it could have been that we missed something. There is a huge number of drugs right now in trial that are based on, if you will, immune or inflammatory pathways. And part of it is we look at the pharmacopoeia of drugs available in all of medicine. There are a lot of drugs like that, and the drug companies are coming to us. The good side of that is there's a couple of good things. One, it's good to have trials. It's good for patients. Just being in a trial, and I'm sure Sabrina will talk about this in a moment, just being in a trial is actually generally good for patients. They do better in trials. So most of them fail. Of course they will. In medicine, 80% of all drug trials that are phase 2 positive fail. But it's an advance. We're doing so much better. We're building new biobanks for biomarkers. All of this in aggregate is very good for patients.
Right. And Jeff, one of the things we're doing at the association is also looking across our portfolio and seeing, do we need to do special call outs for mitochondrial based approaches or endoplasmic reticulum based approaches, right? Or lysosomal based approaches. And so this way we can, even through our funding, start to have an effect on the type of pathways that are being explored. Now that you brought up trials, let's maybe switch gears a little bit. Sabrina, I'll come to you on this is, how do we make trials faster and efficient? We just talked about faster target ID and target validation on the preclinical side. So on the clinical side, how do you do that? And, you have a blueprint that you have been co-leading around the Healy platform trial. So would love to hear, how do we think about faster and efficient trials?
Yeah, no, I'm happy to talk about that. I also was intrigued by many of the comments that Jeff made, and I agree with everything you said, and we could continue to develop that line of thought. But with relation to trials, I think that the most important part of a trial is that it should be designed to provide a decisive answer. So for example, if we're testing multiple drugs that work on inflammation or neuroinflammation, we should be able to, at the end of the trial, to either rule in or rule out a certain target or pathway. So I think that the problem I know, Jeff, you had a little bit of a history, you gave a little bit of an overview of, sort of the history of trials in ALS. And the problem with some of the initial trials was that, again, they wouldn't provide a decisive answer because maybe we didn't have the biomarker. We were not sure if the drug didn't work for one reason or another, not necessarily the target itself. And so now over time, and those were the first trials, and that's fine. When you start doing trials in any new field, really, there were no trials before, and now there were a few, and then we continue to perfect them. And I think that now the contemporary trials are more sophisticated because they can provide a definitive answer about specific drugs, but also specific pathways. And so by process of elimination, if you will, we can also rule out some aspects of for example, a certain class of drugs or subclasses, right? Inflammation is a big term, or autoimmune drugs are a big term. They actually have, there's many different subgroups there. So with respect to that, what I have been working on the clinical research side of things, in collaboration with many colleagues, is to make the trials better. Right now, we're testing 5 drugs, and we're adding a couple later this year. And so the idea is to create an infrastructure that includes those, best practices that as a field we have developed over the last few years, so that we can actually test drugs in an efficient way. And I would say that the platform trial itself, which is, drew inspiration from oncology, because certainly in cancer they are ahead in terms of the efficiency of trials. But we brought that concept to ALS, and now with the Healy ALS platform trial, we have this infrastructure that again includes many of the needs of the community, both in terms of the scientific community, collection of samples in a certain way, sharing of samples in a certain way, again, to spread the knowledge, acquire knowledge and also share it. And also a lot of other needs of the industry partners for robust trials that can provide a definitive answer, needs of the patients to have an accessible trial. So there's many aspects to that. So the trial itself cannot be a cookie cutter, right? We had to incorporate a lot of the knowledge that we accumulated over the years.
Yeah, and Sabrina, I really like what you said. It's not just faster and efficient, but decisive trials. We need to have an answer at the end of that trial. Did the drug work? Did the drug not work? So Jeff, this is one of your topics that you like to talk about quite a bit, about target engagement markers, knowing if you're actually testing the mechanism of your drug in the clinic. Would love to hear your thoughts about this.
Yeah. So again, a little quick history. And Sabrina's right. The trials in the 90s, even 2000, this has been a wonderful evolution of how we do clinical trials, because in the grand scheme of what we do in ALS, and for that matter, any disease, if you have elegant preclinical science, laboratory science, and by the way, it's not always elegant, but let's assume it's elegant. If your trial, the ultimate experiment, is not well designed and well done, what a waste. You've got a great idea, you think you've got a great idea, but you do the human experiment and it's poorly done. And in retrospect, and it's really more of an evolution, the trials that we did a long time ago, they were small. A company would not always listen to us because it took time. And today, what Sabrina described to you is those trials, and when companies come to us, we actually have a panel as part of NEALS, the Northeast ALS consortium that we have, where companies come to us and begin to describe what they want to do. And they're now asking our advice. And we sort of tell them, well, good idea, interesting drug, but you got to do X, Y, and Z. You got to figure out if it gets into the brain. And if you don't, you can do what you want. Now, that's not going to be a good trial, and you're going to probably waste money, and you're certainly not going to help patients the way we think an intellectually well-designed trial should be done. So target engagement is really, really important. One thing I'll point out, though, and second, and after that, is so it binds its target, whatever it's supposed to do. And can you show that it's affected the disease cascade? Something that's supposed to tell you it's mitigating the injury of the disease. That's really nice, too. We have early markers. We have something called neurofilament L that tells us, well, when a cell dies, it releases this. Maybe we can slow that down. And on the horizon are real markers that teach us about that injury cascade. One's called TDP43. We can talk about that if we want, but it is one of those markers. This comes back to the very platform we started with in today's discussion. Those iPS cells, they tell you not only a potential pathway, but you can test your drugs in these living cells from the same patients. And when you do that, not only can you show if the drug is working at its target engagement, you can actually have a readout to say, well, what's changing? And whatever changing, because that maybe I can measure that in a patient, their blood or cerebral spinal fluid through a spinal tap. So you get both the marker of drug efficacy as well as the evidence that it's working at its target. Yeah. So that's exciting for the future.
Yeah, maybe one quick thing, Jeff and Sabrina. We talk about trials and more trials, but more trials means we need more people in the trials. And how do we get more people in the trials? How do you both, as clinicians, talk to your people with ALS, patients who come into your clinic? How do you talk to them about genetic testing? That's not always an easy conversation to have.
Yeah, no, I mean, it's still a complex conversation. I think that what's most important for me as a clinician is to share the availability of targeted products or investigational products that could be applicable to that person should the test results be positive. So I think that that's really what has really changed over the last, let's say, couple of years. Because before then, quite frankly, it was still a complex decision, with no direct applicability. Right now, the situation has really changed over the last couple of years. And so I think that now the conversation is also about the possible access to investigational products that are in development for specific mutations. Now, it's not for all mutations, and some of them may work and others may not. So it's still at the experimental stage, but certainly, this change in terms of the availability now of targeted trials has really completely changed the conversation in my mind.
Yeah, I completely agree. And my conversation, first of all, every patient I see is now offered genetic testing. Obviously, it's up to them. I never, of course, force it. And then I divide it in a different way, the three different bins that Sabrina just mentioned. One, it's potentially diagnostic. It depends when a patient gets to our clinics, how early or late in the disease is. Secondly, it's exactly what Sabrina said too, which is that it makes them eligible for exciting trials, gene therapy. Not all, but if we don't know their gene mutation, if they have one or not, we need to know that for trials. And lastly is the family issue. That's actually one, at least the way I practice, I often let them know there's a possibility of how it can be important to the family. But generally in my clinic, if they then want, maybe a spouse is there or a child's there, I bring in genetics counselors for that phase. I'm not a genetics counselor, but I can talk, the patient's my patient. I can deal with the initial issues, diagnosis and trials. And then we usually bring in a genetics counselor for the last bin. But the value of that is enormous. And to be fair, I don't know the exact number, but at least 80% of patients, maybe it's closer to almost 100%, now get these free panels, and the fact that they're free is fantastic. I can tell you years ago, they were a fortune, but it also means I'm finding genes that I would have never found before in patients. I'm finding patients who have mutations that account for their disease and it just wasn't obvious. And that's really important. It's not important for them, for future drugs. And just to add to that, especially when it ges to family, as you know, and of course, Sabrina knows, at least some of the mutations, and now I'm speaking of the SOD1 mutation, we're already into the next generation of medicine, which is to say, someone carries a mutation, they're not ill yet, we're going to be following them. And as soon as there's a hint biologically, not even clinically, biologically, if there's something wrong, we could institute therapy. That's getting in early. And that's what one day we have to do with all of our patients, get in as early as possible. And it's genetics that's allowing us to do that.
Exactly. And, this is one way to also increase the number of people, the pool that's available for clinical trials, because as we know, later diagnosis means maybe they are now no longer, in the inclusion criteria. And so they can't, even if they want to, they can't participate. So I'm glad that you brought up that. So, maybe we switch gears, Sabrina and Jeff. And
before we do, Kuldip, I want to bring up one more thing. In terms of increasing patients, I literally just had this conversation the other day. We do have set criteria. We as a community, and of course, the whole point of a trial is, can we prove in the time frame of the trial that we've done something to slow the disease down? That often means we exclude a fair number of patients. Of course, Sabrina knows as well. But early trials where you're just trying to figure out, does the drug work at all at its target? This gets back to the target discussion. So not clinically, but you need to first know before you even get to an efficacy trial, does it work at its target? Those patients don't have to be so restricted. You can take patients that, what we call slow patients who've had the disease for many years, who are not eligible usually for trials. But if we have those biomarkers, this gets back to the TP43, and we can show that there's a pathway in them. It's an injury pathway. Wow, you have a reporter for, does my drug affect that pathway? They may not show clinical efficacy, but your drug's affecting the pathway. And that opens the door to many, many more patients who at least could be in phase 1 trials. Yeah. So I would argue, I'm throwing this to Sabrina.
Yeah, and actually, I wanted to expand on that. I completely agree. I think that this also goes back to the point of collaboration between academia, the patient community, and industry. Sometimes trials can be designed as sort of cookie cutters, right? Like kind of, if that's always been done a certain way, you see how inclusion and exclusion criteria are, and they can be repeated. Instead, I think we have learned as an academic community to really be flexible and really use certain criteria for the right phase of development.
Well, thank you both. What I was going to say, something Jeff said earlier about that interaction with the person with ALS and their family. I wanted to touch on sort of the care part of this, the managing ALS today. So we talked about drug discovery, and that's for, in the future, we'll have a drug, but there are people living with ALS today, and there are just so many decisions, as Jeff, you and Sabrina just talked about, that you all have to make in real time, looking at where the patient is in the disease journey. When to provide respiratory intervention, when to provide GI intervention, and so can you, Sabrina first, and then Jeff, can you talk about sort of the concept of a multidisciplinary clinic, and why is that so important? How is that helpful for the outcomes for that patient?
Yeah, both Jeff and I work in multidisciplinary clinics, and at this time, there's several clinics. I think there should be more, but certainly there's many clinics in the US and globally. They really specialize in the care of the person with ALS, and that it's always a collaborative approach, because ALS affects many areas, obviously many regions in the body, and requires specialized expertise to really maximize outcomes. It's been shown many times that attending a multidisciplinary clinic improves outcomes. I would say that while I love clinical trials, and that's what I do most of the time, I never forget, and in fact, I always emphasize with my own patients I see in clinic, that we already have interventions that are proven to be effective and prolong life and survival, such as, for example, initiation of BiPAP when it's important to do so. And for sure, we have several interventions that can improve quality of life, quite frankly, much more than in a drug that we don't know if it works yet. So, and the reason I emphasize that is because, unfortunately, sometimes, again, the importance of multidisciplinary care is not as emphasized. And so it's really, really critical that we give all our patients access to the best possible care, because otherwise, we're missing out on things that are already available and should be available to all.
Yeah, I couldn't say any better. That's so true. And the other point to this, and at least for me, if I'm honest, I'm a neurologist. I'm not a pulmonary physician. I'm not a nutrition specialist. So you need, and since patients don't have they come to us with neurologic problems, but the manifestation on their body are many, many systems. They can't eat, they can't take the proper nutrition, they can't breathe properly. And so we need those experts in different areas. In fact, I often tell my patients, fine, you've met me, we're going to talk about what's going on in you and make the diagnosis. And now I'm going to hand you to my team that's really going to keep you in the best shape possible, which are those other physicians and practitioners, occupational therapists, physical, everything that Sabrina just mentioned. And that is the best care. I often use the analogy with patients. I tend to use cars. Patients aren't trained in medicine. So we think of the Indy 500, you got this fantastic race car, but it's got to go to the pit crew where everyone really works on that car to keep it in the best shape. Our clinics are those pit crews to keep patients in absolutely the best shape possible.
Yeah, thank you both. And, I've been thinking about this in terms of the role of the multidisciplinary clinics is going to be even more important when a drug is actually approved. How do we, both from an infrastructure standpoint, but also from an operation standpoint, make sure that that clinic continues to provide better care, optimal care, while you have new drugs coming on the market? So this is really exciting. Jeff, were you going to say something?
Yeah, buried in what you just said is so right. Sabrina and I, big medical centers, some of the biggest in the U.S. that care for ALS, we're experienced, but there are a lot of patients out there, and what is lacking is that kind of care in the vast majority of the United States. And so one of the challenges in the future, especially if we have things like this, is when patients go back to, I'll say this, rural America, how do we make sure that they get that kind of care? Because they're not all going to be able to take 5-hour drives into their local city. And I don't have that answer, but that's something we certainly have to pay attention to. There's something buried in what I just said, though. There is something that's come out of the bad of COVID, and that's we all got used to telemedicine. We all got used to taking care of patients that couldn't always make it to clinics. Not necessarily, it's not, in my view, it's not the best for making initial diagnosis. You need hands-on, in my opinion. But once you have established that relationship and maintaining contact with patients, telemedicine is really quite important. And secondary, that's not really secondary, is the use of tools like apps, smartphone apps, and devices at home that can actually continue to measure patients' physiology, how they're doing, and feed that back to us so they don't all have to drive hours to come to our clinics, but we stay in close contact with their true neurological status and then what we can do from afar.
Yeah, that's so important. And I think that will require, hopefully soon, if we have many drugs approved, some significant changes to the way healthcare is delivered for ALS. So I think there's going to be a need to better support and resource both the big centers that have already existing clinics, as well as their connection to maybe more remote areas where maybe it doesn't make sense to have an ALS clinic because the area is remote, but there should be an ability to connect with telemedicine or wearables or other tools, as you mentioned. I think this will certainly require some changes also in their reimbursement practices and sort of the way clinics are resourced.
Absolutely. Well, I think our time is coming to an end here, Sabrina and Jeff. Maybe just if all three of us could probably end on, sort of the optimistic, where do we see us in 10 years? I mean, I'll tell you what I'm really excited about. I hope I'm not stealing what you were both going to say, but we have never had this many ALS trials in the ecosystem. I mean, who knew for a rare disease like this? We're looking at over 60, 70 trials worldwide between phase 1, 2, and 3. And that's a really exciting time. And what's exciting is that it's different type of approaches. So there's small molecules, there's antibody approaches, there's stem cell therapies, and there are different targets and different pathways that people are going after. So there are big companies and there are small companies. And so that's what makes me really excited. I think the concept of can we on the preclinical side validate fast? On the clinical side, can we test rapidly and in a way that informs how the drug works? And then on the care side, how do we make sure that when a drug is approved, that's incorporated into the clinic? And so that's what I'm excited about. And so, Sabrina, maybe any last comment for you?
Yeah, no, I completely agree. And I would say, and I'm sorry, Jeff, but I'm luckier than you, because I got into the field at the time of exponential growth. So you started it, and now I'm benefiting from that. And so, over the last 5-10 years when I've been in this field, I myself have seen this exponential growth. So I'm very optimistic, I have to say, because even if I think about, actually, the other day I was looking at some of the initial slides, when I finished training 10 years ago, the field was so small, so much smaller, and few trials, and now it's really like exploded. So this is the right time for ALS. Yeah.
How can I not agree with all of that? So there's a couple other things. So one, no question, I actually believe in 10 years we will have more drugs. I don't know how many, I don't know how powerful they'll be, but we will likely have more. I think we'll have a better understanding of targeting subgroups of patients, meaning sporadic patients. Or at least I'm hopeful of that with today's tools. The other thing, buried in actually what Sabrina just said, and it's true. I have lived through a lot of that science, from the first discovery of SOD1 to the early mouse models, the early trials, the gene therapy trials. One of the big changes, it's not just that there are more companies, there's more of us. They're more intelligent, committed, both clinicians and scientists. That community is huge.
Well, thank you. I want to, again, thank the WuXi AppTec team for asking the three of us to be on this panel and share our thoughts about the future. We hope that in 10 years, this disease is a livable disease, and that we have multiple treatments, that we don't have to even be afraid of telling someone that they are diagnosed with ALS, that they can live their life, albeit with treatments, but that their lives are extended. So thank you to the team, and Sabrina and Jeff, thank you so much for joining me on the panel.
Thank you.
My pleasure. It's been great. Thank you.